So the sixth and final topic that we have to cover in biochemistry is biochemistry and the environment. And a lot of the ideas in this subtopic are pretty common sense, although there is some vocabulary that we need to go through in order to understand the main bullet points that IB wants you to know. The first vocabulary word we need to have is xenobiotics. Xenobiotics are chemical compounds in a living organism that are foreign to that organism. They should not be there. Or it could be a chemical that does belong there, but not in such high quantities as we're actually finding it. Xeno means foreign, so these are foreign to the biology of the animal. And some examples of xenobiotics are drugs, antibiotics, hormones, things like that, insecticides, heavy metals, plastics, and food additives or other pollutants in the environment. Xenobiotics can have different effects on organisms depending on their properties. Polar molecules are usually not such a big problem because they can be flushed from the system, excreted in the urine, removed from the body. Non-polar molecules enter cells, passing across that hydrophobic cell membrane. And once they're in there, best case scenario is that there are enzymes in the cell that can somehow modify or detoxify this xenobiotic substance. Although that sounds like a great thing, sometimes the reason we've used the chemical is because we want the cell to be destroyed. We use pesticides on purpose to kill insects. We use antibiotics on purpose to kill bacteria. And if those insects or other pests can detoxify the poison that we're trying to give them, then they can build up a pesticide resistance. Microbes, bacteria that we put antibiotics on, if they can detoxify those antibiotics, they build up a resistance, and then using those chemicals becomes pointless. On the other hand, if the cells cannot produce enzymes to kind of detoxify the substance, then you can get a buildup of these xenobiotic molecules in cells, and this is called bioaccumulation. For instance, tetraethyl lead can accumulate in fat tissues, and then when you have this buildup over time, it can have toxic effects. So one example we're going to look at in the environment in terms of xenobiotic substances is on the next slide. We're going to start talking about some pharmaceuticals. So pharmaceuticals are things like antibiotics, like painkillers, like hormones that can be flushed from the body through the urine, but then that urine, those pharmaceuticals don't get broken down completely in waste treatment plants, and they end up in rivers and lakes where then fish and other aquatic organisms can be exposed to those pharmaceutical compounds. So, for instance, there are synthetic estrogens in wastewater from urine of women who are taking contraceptive pills. Also, there are xenoestrogens, foreign estrogens. These are chemicals used in industry that imitate the effects of estrogen. And some examples of xenoestrogens are PCB, polychlorinated biphenyl, which was used in electrical components until 1979. And more recently, bisphenol A, BPA, is used to make plastics, and there are ongoing debates about safe levels. But right now, you see this is BPA free on lots of plastic items that you can buy in stores now. The concern with synthetic estrogens and xenoestrogens, for the concern with both of those substances, is that male fish may become feminized and unable to breed. So the concern in the environment is that this can affect organisms that are living in the lakes and streams where these synthetic estrogens and xenoestrogens are flushed into. Biomagnification is an increased concentration of a xenobiotic in higher trophic levels. For instance, DDT is the most commonly used example of biomagnification. DDT was a chemical used to control mosquitoes responsible for malaria and typhus. It's fat soluble, it's not metabolized by organisms, and it was banned in the 1970s because people finally noticed that birds of prey were declining due to their fragile eggshells. And the reason this happened was because DDT in the environment was taken up by very small amounts in plants, aquatic plants. But then the tiny aquatic animals that feed on those aquatic plants have to eat a lot of that plant in order to stay alive. And so even though there was just a tiny amount of DDT in the phytoplankton, this was concentrated, it was magnified in the next trophic level that needed to eat the phytoplankton. Then as we moved up the food web, up the food chain, the fish that eat the zooplankton ended up with an even higher concentration of DDT in the system because they have to eat a vast number of zooplankton in order to stay alive. And then even larger predatory fish needed to eat large quantities of the small fish in order to stay alive. So there was even a higher concentration of DDT there. And finally, when you got to these fish-eating birds, the concentration of DDT in their bodies is extremely high. And this DDT caused fragile eggshells, and then just the act of mothers sitting on their eggs to keep them warm would break the eggs, and baby birds stopped being born. Biomagnification does not just happen with DDT, just DDT is the most common example. Biomagnification can happen with dioxins, with PCBs, with heavy metals, and this is why, with heavy metals, this is why you should not eat too much salmon, tuna, swordfish, because those are large predatory fish where there will be a higher concentration of the heavy metals in their bodies. It is important to note the difference between bioaccumulation and biomagnification. Bioaccumulation happens within one organism. A chemical, a xenobiotic substance, builds up in the cell of one organism. Biomagnification happens across a food web, where substance builds up in higher predators because those higher predators need to eat a vast number of smaller organisms in order to stay alive. So how do we deal with xenobiotics? Amelioration is the response to xenobiotics. Amelioration are ways that scientists are coming up with to lessen the problems of xenobiotics or to improve the outcomes of xenobiotic chemicals in the environment. There are four main strategies for dealing with xenobiotics in the environment. Those are using HostGuest Chemistry, using biodegradable substances, using bioremediation techniques, and using green chemistry strategies. And we're going to look at each of these four in turn. First, looking at HostGuest Chemistry. HostGuest Chemistry is kind of a man-made version of enzyme substrate chemistry. Just like in enzyme substrate chemistry, you have an enzyme, you have a substrate, and those two can bind reversibly together to make an enzyme substrate complex. Here we have a host molecule that's man-made, specifically designed to attract and bind reversibly to a guest molecule, which is the xenobiotic substance. And when they join together, they form a supermolecule called a host-guest complex to trap the xenobiotic substance. Just like with enzyme substrate chemistry, just like how the substance substrate binds non-covalently to the enzyme. The xenobiotic guest molecule also binds non-covalently to the host molecule to form the supermolecule host-guest complex. Each host is specifically designed to capture a specific guest, just like each enzyme is specific to a particular reaction. Each enzyme needs to have a very specific shape to bind reversibly with its substrate. We need to have the chemistry of the host just right to attract the xenobiotic guest substance. What kind of bonding holds the host and guest together if it's not covalent interactions? It's the same things we saw in protein tertiary structure. With the enzyme substrate complex. We've got hydrogen bonding, we've got ionic bonding, we've got hydrophobic interactions, pretty much everything except for covalent bonding. The host structure often has some sort of cage or capsule or tube design, like you can see here where the guest molecule is kind of trapped inside of this cage. And these cages and capsules you actually saw examples of in topic 4 with chemical bonding and structure, where we looked at buckyballs and graphene tubules and things like that. This is one chemical application of those large carbon structures that we saw in topic 4. The next amelioration method that we're looking at is using biodegradable substances. Biodegradable substances are those chemicals that can undergo natural degradation into non-harmful end products. And by natural, we usually mean from bacteria. The hot topic for biodegradable substances is biodegradable plastic. There are two main types of biodegradable plastic, and we'll look at each one. There's plant-based hydro-biodegradable plastic, hydro meaning water, and then there's petroleum-based oxo-biodegradable plastic, and that's through oxidation processes. So let's look at these plant-based hydro-biodegradable plastics first. These are usually starch-based, often made from corn. And the breakdown is initiated by hydrolysis. Okay, and these hydrolysis reactions, this produces carbon dioxide and water, and these decompose quickly at high temperatures. In a landfill, these may decompose more slowly and may release methane. Both carbon dioxide and methane are greenhouse gases. These plant-based biodegradable plastics may be compostable, so you may be able to add them to your compost heap with your garden waste to break down naturally there. The other kind of biodegradable plastic is petroleum-based, and it's oxo-biodegradable. And what happens with these plastics is these are derived from oil industry byproducts, and then some kind of additive is added to the plastic to catalyze the breakdown into microfragments. So you can see here we started off with an intact plastic bag. After 30 days, you could see a noticeable difference of kind of the plastic breaking down into smaller shreds. It's not disappearing. We've got shreds here at the bottom of the bag, and after 55 days, it's broken down into these multiple tiny shreds of plastic. The idea is that these additives break the plastic down into small enough pieces that can then be processed by bacteria. So this is oxo-biodegradable plastic. Bioremediation. Bioremediation is using microorganisms to help break down xenobiotic chemicals. The main thing that you've probably heard this used for is managing crude oil spills. Crude oil contains many different compounds, so many different bacteria or many different fungi are needed in order to break down the wide variety of chemicals that you have in crude oil. Larger and branched hydrocarbons are less likely to be broken down. They're harder to break down. And the problem with managing crude oil spills this way is that the whole process may just take too long. Ecological damage may occur while we're waiting for the bacteria to work. So seabirds are getting covered with oil and things like that and dying while we're waiting for bacteria to work. So this process isn't perfect, but scientists are looking at ways to make bioremediation work faster, particularly in the case of oil spills. Bioremediation is used in other industries, for instance on industrial waste like in paper mills or textile mills or leather mills, to break down the chemicals that are used in those factories. The final topic that we have to talk about is green chemistry, and green chemistry can also be called sustainable chemistry. Sustainable chemistry or green chemistry seeks to reduce environmental footprint of chemical manufacturing by while still improving product and environmental safety. There are 12 general principles for green chemistry. You don't have to memorize these, but your textbook mentioned them, and I thought it was mean for them to mention them and then not tell you what they were. The general idea is to reduce waste, to use less hazardous chemicals, those sorts of things, to reduce the environmental footprint of chemical manufacturing. So some examples of how green chemistry is being used. Toxic solvents used to be used to kind of decaffeinate coffee, and through green chemistry we've discovered better methods like using pressurized carbon dioxide to decaffeinate coffee. We can use enzymes instead of having to use harsh chemicals or high temperatures for various reactions. For instance, esterification reactions for face creams, we can use enzymes for that instead of using high temperatures and concentrated sulfuric acid. We can add enzymes to laundry detergents, I don't know what to call them because we're using them instead of detergents, to laundry cleaning products instead of detergents to clean clothing, because the phosphates that are present in detergents can cause algal blooms in rivers and lakes. So reducing the amount of detergent needed to clean clothing by using enzymes. Green chemistry is also responsible for helping us source renewable textiles like bamboo and eucalyptus. So here are some practice questions to help you think about biochemistry and the environment. And I hope this has helped you.